Hi everyone. Today let's move on to yet another concept in genetics. It's related to the chromosomal theory of inheritance, which we had already completed in the last video. So in that chromosomal theory of inheritance, we learned regarding the theory that chromosome is the basis of the genes. That is, chromosomes are the vehicles of genes, or chromosomes carry the genes. And I also told you that in the next video we will be learning regarding the experimental proof for the chromosomal theory of inheritance. So today I have come with that. That experimental proof for chromosomal theory of inheritance actually gave birth to two new concepts. So what are they? The two new concepts, linkage and recombination. The two new concepts are named as linkage and recombination. This is actually a continuation of chromosomal theory of inheritance. The chromosomal theory of inheritance, that theory was actually taken for verification by a team of scientists which was headed by Thomas Hunt Morgan. He is called as T.H. Morgan. So Thomas Hunt Morgan, or T.H. Morgan, and his colleagues were actually working on chromosomal theory of inheritance to find out the experimental proof for that. As we know, to carry out an experiment, we have to first design an experiment. So Thomas Hunt Morgan and his colleagues designed the experiment, or they took the experimental material, not that pea plant or things like that, but on fruit flies. I hope most of you might be knowing small flies, fruit flies. Its scientific name of fruit fly is Drosophila melanogaster. Scientific name is Drosophila melanogaster. Once again, Drosophila melanogaster. That is the scientific name of fruit fly. So even if it is asked for the examination, when you write the name Drosophila melanogaster, be very careful. The genus name starts with capital letter, species name with small letter, and both the genus name and the species name have to be underlined separately. So if it is asked as a 1-mark question for you for the exam, also give the scientific name of Drosophila the scientific name of fruit fly. You will be writing the correct spelling and everything, Drosophila melanogaster, correctly. But if you forget to underline it separately or start the name, genus name with capital letter and species name with small letters, such small things, you have to keep it in mind, because that is the rule for writing scientific name. So since you are in +2 or in the higher classes, we should know that rule when we do it. Okay, so accordingly, Drosophila melanogaster, scientific name of fruit fly. So Thomas Hunt Morgan and his colleagues worked on Drosophila melanogaster to frame an experimental basis for the chromosomal theory of inheritance, and in turn they framed they gave the experimental proof, and in turn they also came up with two new concepts, linkage and recombination. So why they took this experimental material as Drosophila melanogaster? There are 5 reasons for that. Why the experimental material was Drosophila melanogaster. Okay, first and foremost thing, these flies can be grown very easily in laboratory by providing simple synthetic medium. Simple medium means the food for them. So with the help of simple synthetic medium, it was very easy for the scientists to grow this Drosophila melanogaster in the laboratory. So that was the first reason. They could be grown on simple synthetic medium very easily. Next reason is their life cycle. These flies, they could complete their life cycle within 2 weeks. So in order to find out the passing of characters from one generation to the next generation, the scientists had to wait only for 2 weeks. So life cycle they could complete within 2 weeks. So it was actually easy for them. It was time saving. So it was the next reason. Third reason is regarding the number of progenies. A single mating could produce large number of progenies. So they could find out the variations in a large number of sample after a single mating. That was also time saving. Next one, males and females were easily distinguishable. The male flies and the female flies could be easily distinguished by just at a sight. We can distinguish male flies and female flies, or with the help of low power microscope also it could be done very easily. So since they were easily distinguishable, again it was a reason for why it was taken as the experimental material. And the last reason was hereditary variations could be observed with low power microscope. See, for that point, let us come to that pea plant which Mendel has taken. He studied 7 different characters, isn't it? 7 different contrasting traits of pea plant. So those 7 different contrasting traits could be easily observed, whether it is flower color, height, pod shape, pod color, seed shape, seed color, all these things could be easily distinguished. Similarly, when this fruit fly was taken, fruit fly also had some characters which could be easily distinguished under low power microscope, and those characters was passed on from parent to progeny. That means those characters were hereditary characters. So in fruit fly there were many hereditary characters which could be easily observed under low power microscope. So because of these 5 reasons, fruit fly or Drosophila melanogaster was taken as the experimental material by Thomas Hunt Morgan and their colleagues for framing the basis for chromosomal theory of inheritance, an experimental basis for that. Now regarding their experiment, we can just focus our attention on 2 different types of crosses. Again, I am comparing this with pea plants so that it is easy for you to understand. In the pea plant, crosses was taken how? 1 character was taken into account, monohybrid cross, then 2 characters were taken into account, dihybrid cross, isn't it? Here, T.H. Morgan carried out dihybrid cross. So in the dihybrid cross, after taking the F1 generation, what is done with the F1 generation? The F1 generation is self-crossed, and after self-crossing, F2, in the F2 the ratio 9:3:3:1, isn't it? This is the thing that Mendel got. T.H. Morgan was also doing the same experiment and was waiting whether he is going to get the same ratio or not. So let us focus our attention to the experiment that T.H. Morgan had done. Here he did dihybrid cross. Dihybrid means he has taken 2 characters into account. Okay, what were those 2 characters? Those 2 characters were body color of fruit fly and eye color of fruit fly. 2 characters, body color and eye color. Now, what was those 2 body colors? Body color was, some flies had brown body color, some others had yellow. So body color is of 2 types, brown, yellow. Like we had in Mendel, height 2 types, tall, dwarf. Similarly, here body color 2 types, brown or yellow. Next, eye color also 2 forms were there. Some of them had red-eyed, some of them were white-eyed. So red eye color and white eye color. So 2 traits, each trait is having 2 forms. So dihybrid cross he is doing. Now coming to the next question. Among these 2 forms for each trait, which one is dominant, which one is recessive? Okay, so here in this particular experiment, instead of the word dominant, we use the term wild type. Wild type means naturally occurring type, occurring in most in number in nature. Wild type, naturally occurring form. So wild type is used instead of the word dominant form. Dominant form we used in Mendelian genetics. So instead of dominant we are using another term here, wild. Both means the same. Okay. So here wild type is brown body color. And recessive. Instead of recessive we use the word here mutant. Mutant means some change has occurred. It is not wild type, but from the wild type, from the naturally occurring type, there occurred some changes. So, in order to understand that some changes has occurred, we are using the word mutant type. So, instead of dominant form, we use the term wild. And instead of recessive, we are using the term mutant. So, there were two body color, brown and yellow. Which one is dominant? Brown is dominant. So, instead of dominant, what word will we use here? Right, wild. So, brown body color is wild type. And what about the yellow? Yellow is recessive. Instead of recessive, what do we use here? Mutant type. So, wild type, mutant type. Wild type is brown, mutant type is yellow body color. Okay. So, here I have written here wild, brown, mutant, yellow body color. Now coming to the notations. In the Mendelian genetics, we used the letter capital and small letters, isn't it? Capital to denote dominant and small to denote recessive. Here, there is no capital and small. Letters are same. Instead, we use a plus sign. When we write plus, means it is dominant or it is wild. If we don't use that plus, it is recessive or it is mutant. Okay, so how do you do that? We take the letter of this mutant type, Y, yellow, Y, not this brown, not B, but instead we take Y, and with that Y, if you simply write Y, it is mutant. If you give a plus to it, it becomes wild type. So which letter we will take? The recessive forms letter here we are taking. The Y, the mutant type. Mutant type is yellow, so Y. If you put a plus sign, what does that become? It becomes a wild type here. Which is the wild type? Brown. I hope this is clear. Now moving on to the next trait. Next trait is eye color. See, eye color, two types were there. Which are they? Red and white. I am saying red is wild, means red is dominant. And what about white? White is recessive, so white is mutant. So which letter will I take? R or W? I should take the mutant form. So W, I am going to take the letter. If I simply put W, it is white, recessive. If I put a plus sign for that, it becomes dominant or wild type or red eye color here. I hope that is clear. So, red eye color red, W plus. If it is white, simply I put W. So, this is wild type red and mutant type white. Okay. So, again, we were just dealing with the notations and all. Again, we will just come to that experiment. Thomas Hunt Morgan and their colleagues were trying to frame a experimental basis for chromosomal theory of inheritance, and for that they took the experimental material as Drosophila melanogaster or fruit fly, and they observed some of the hereditary traits. Among that, in one particular cross they took two hereditary traits, that is they were doing dihybrid cross, and the two traits taken were body color and eye color. Body color brown is dominant or wild over yellow, and eye color red is dominant or wild over white eye color. Okay. Now let us come back to the cross. Here, you can see here I have written yellow white. That means yellow white I have written here, yellow bodied, white eyed. So imagine a fruit fly having yellow body color, white eye color, and that fruit fly is female. So that fruit fly is totally mutant or recessive, isn't it? Fully recessive for body color as well as eye color. So yellow bodied, white-eyed female is crossed with totally white, that is brown-bodied and a red-eyed male. Are you able to imagine? See, imagine a yellow-bodied, white-eyed female fly is crossed with brown-bodied, red-eyed male. They are crossed. Here it is not like pea plant pollination we cannot do. They are cross-eyed. They are crossed here, and see, I have put here sign. This sign is female and this sign is male. So mutant female and wild male are crossed and waiting for F1 generation. How many days? 2 weeks. Life cycle is 2 weeks. F1 generation, what did they get? Females were wild. Females were wild means females had brown body and red eye color. This is wild, brown body, red. Females were wild type and males were mutant type. So from this, they were again waiting. After F1, what's the next thing? Self-crossing of F1. So F1 progenies, males and females, whatever they got, those were intercrossed. So here we cannot use the word self-cross. Instead, we are using the word intercrossed. So these are the terms which you should be very careful while writing your answers. Don't write self-crossed but intercrossed. Okay? F1 progenies were intercrossed and we are waiting for F2. Plenty of F2 progenies. Then we started counting the numbers. How many of them were like their parents? How many of them had new form? Majority of them, that is 98.7% of the F2 progenies were like their parents. Means whether males or females, they had yellow body, white eye color, brown body, red eye color. Majority of them. But there were a few, very less than 2%. That is 1.3%. They had a combination. What kind of combination? They had yellow body color but red eye color. See, one type is one form, that is body color is mutant, eye color is wild. Are you able to imagine? Few of them had a combination. Yellow body color, red eye color. Another few had brown body color, white eye color. So that was a combination was different. So you call them as recombinant, recombined forms or new forms were occurring. So how many new forms? Very few, less than 2% or 1.3% only recombinant. Rest 98.7% of them were parental. Parental means what? If the body color is yellow, eye color is white. If the body color is brown, eye color is red. This combination was there. Whether males or females, they had this combination only. But very few had another combination, that is yellow body color, red eye color. Similarly, brown body color, white eye color. This combination was very less. So they were thinking, why this few new combination had occurred? What is the reason behind recombination? To find out that, they were actually finding out the position of these genes on the chromosome. So these genes, that is genes responsible for body color and eye color, were present on a chromosome called as X chromosome. Okay. Gene responsible for body color and eye color were present on X chromosome. Where they are present? In order to find out new and more clear clarity for that, they did another type of cross as well. Now the second cross. So first cross we have completed with body color and eye color. Next set of cross is with wing size and of course eye color. Wing size and eye color. So eye color we have already dealt with, isn't it? Red and white. Red is wild over white. Or red is wild and white color is mutant type. Now the next thing is regarding wing size. Large winged fruit flies were there. Fruit flies were also there which had very small wings. Instead of small, we call here the word miniature. Miniature. Very small. Miniature. Okay. So here, wing size, two types, large winged and miniature winged. So among these two, which one is dominant or which one is wild? Large wing size is wild. So which letter will we take? We will take of the other form, isn't it? Mutant form. That is miniature. So here we take the letter M to denote the wing size. If you put simply M, it is miniature. If you put a plus sign for that, it becomes mutant or it becomes wild type, not mutant, wild type or large winged. So miniature denoted by M, and if it is M plus, it is large wing sized. Okay. So when Thomas Hunt Morgan took two fruit flies, male and female, and always he used to take male flies with wild character and female flies with mutant character. Okay, so in the last cross also we have dealt with that females were yellow bodied, white eyed. They had a yellow body color and white eye color. That is mutant. Crossed with males which had brown body color and red eye color. So males were wild type. Here also males had wild form. That means they had large wing size and red eye color. So males red large. Red large here, whatever I have written here, red large means they had red eye color. Imagine that, fruit fly having red eye color with large wings. Okay, here two characters are only taken. They were crossed with females having white eye color with miniature wings. White eye color with miniature wings. So mutant females were crossed with wild type males. What is F1? F1 wild females and mutant males. Next, what is the step? They were intercrossed. F1 progenies were intercrossed, waiting for F2. In the F2, what they are looking for? They are looking for the percentage of fruit flies which looked exactly like the parents and another category percentage which had a new combination. So how many? In F2, 62.8% were parental type and 37.2% were recombinant type. Here if you compare, parental type is more than recombinant type. It is true. In the last cross also, parental type was 98.7%. It was much larger than the recombinant. Recombinant was 1.2% only. But here see the recombinant, 37.2%. Okay. Now let us place these two results in a tabular column and try to find out some inferences. Here it is. The results. Traits for the first cross: body color, eye color. Parental type was 98.7, recombinant type 1.37%. Now, when you look the next one, wing size and eye color, 62.8% parental, 37.2% recombinant. Here, of course, parental type is always more than recombinant type. But when you compare the parental types, the parental type for body color, eye color is more than that for wing shape, wing size and eye color. When you compare these two characters, that is body color and eye color, is showing more occurrence. Parental type is more and new combinations are very less there. But when you take the other thing, wing size and eye color, new combinations are coming. What is the new combination? See, parent that we have taken is white eye color miniature wing. That means imagine a fruit fly having white eye color and very small wings. Okay, that is one character. Another character, they had large wings and red eye color. So these are the parental characters. These parental characters were there in the F2 generation also. About 62.8%. 62.8% of the fruit flies had this character. But 37.2% had recombinant character. What is the recombinant character? They had small wings but not white eye color but red eye color. That is new combination. Here small wings, white eye color is parental type. New combination means small wings with red eye color. Similarly, large wings with white eye color. So that kind of combination also came. But that combination was less than parental type but more than the previous cross. These are the results. Now what is their inference? What did they infer from this? They inferred that these genes are responsible for these traits, genes responsible for wing size, eye color, body color. For each trait, I told you there is a gene responsible, isn't it? And for these three traits that they have taken, there are genes responsible. These genes are present on chromosomes, or chromosomes are the carriers of genes. That is chromosomal theory of inheritance, right? Now we are actually coming towards chromosomal theory of inheritance and Morgan's experiment. So, according to chromosomal theory of inheritance, genes are present on chromosomes. So, for each of these three traits that they have taken, those genes are also present on chromosomes, right? Now, where on chromosomes they are present? What is the relative distance? That was the question. Now, these genes, for each of these characters are present on the X chromosome of fruit fly. Fruit fly is having many chromosomes. Like we human beings are having 23 pairs of chromosomes. Like with fruit flies also. But these characters, the genes responsible for these characters are present on their X chromosome. All these three genes are present on the X chromosome. Then why this difference? Then why this difference of percentage in parental type and recombinant type? Here is the answer. See here, we first understand this that I have drawn here, this denotes two X chromosomes. This denotes one X chromosome and another Y chromosome. So X and Y, if it is coming, this is male, and XX denotes female. So this is the first cross that we have taken. Yellow body color, white eye color. So X, in the X chromosomes they are present. Females, they were mutant, so there is no plus sign here, simply Y and yellow. X chromosomes, these are the genes responsible, so I have drawn it on the X chromosome. Female is what type? Female is mutant type, so simply you have to denote by Y and W. It is crossed with male, so to denote male, one chromosome is X, another chromosome is Y. So I have changed the shape and I have drawn like this. In your textbook also you can find this. So X and Y. So if it is like this, two chromosomes different, X and Y, two different shapes, this male and this is female. Okay, female is mutant. What about male? Males, Morgan has always taken males as wild type. Wild type means we should denote with plus sign. So here I have denoted Y plus W plus. These genes are on the X chromosome. These are the X chromosome, this is X chromosome. And on the same position I have drawn. Now the next cross. Next cross is which two things? There is no body color. But instead of body color, what is taken? Wing size is taken. So gene for wing size is not present here. This area is for gene for body color only. But gene for eye color is present here. White eye color. White eye color. Okay, it is denoted by W. What's the other thing I have taken? Eye color and wing size. So wing size is not here. Wing size is somewhere away from here. So wing size is here. The gene for wing size is present on X chromosome at this place and this area. If I am drawing here, means this area is for body color. So first cross is with body color, eye color. Next cross is with eye color and wing size. Eye color and wing size. So wing size gene is present here. So this I have drawn the same type of chromosome. So this is XX female mutant crossed with, see, white miniature wing mutant female crossed with what? Red eye colored fruit flies having large wings. So red eye color means wild type, so W plus. That is why I have not shaded this area to show this is another form. And M plus, I have not shaded here. You can see here I have shaded this area, not this area. Okay, so W plus, M plus. And this is Y chromosome, so this is male. They are crossed. So here, why this huge difference? That was our question. The huge difference is because though these genes are there on the X chromosome, in the first case these two genes are very close together. The second case this is far apart. There is more distance between the genes. Which two genes? Genes for eye color and wing size. But for genes for eye color and body color are placed close together. Genes for eye color and wing size are far apart. Since the genes are far apart for eye color and wing size, there is more chance that they can combine in different combinations. That is, here chromosome can break, here also this chromosome can break, and M plus can come here. M can go there. What is it? Breaking and joining? This breaking and joining takes place during the meiotic phase of cell division. I hope you have learned in class 11 regarding two types of cell, the karyokinesis, that is mitosis and meiosis. In meiosis, it is especially done for gamete formation. It is reduction division where new combinations are formed. During packaging, crossing over takes place. That crossing over is breaking of chromosome and joining with another region of chromosome. Breaking and joining. That breaking and joining, in that process, when the genes are together, when they break and joining, chances are there that they go together. They can break here. When they break, they go together and they join with another one. When they join with another one, yellow body color, white eye color, it goes together. But here when the break happens, see, here when the break happens, this eye color and wing size, they are far apart. So they cannot go together. One can go independent of the other. But here, more chances are like whenever they break and they go together. That is why more of parental type is here. Because they are going together. As in the parent, they are going in the progeny. But here, when wing color and eye color is taken, since they are far apart in the chromosome, it is always not necessary that They will break and they will go together. They can break and they can go as two independent pieces. And they can have new combinations. As they can recombine. They can recombine and have new combinations. I hope you are getting my point. So, in nutshell, when genes for characters are together, they are linked. They are together, they are linked. Linkage. When genes for a character are far apart, they can have recombinations, new combination. That is this. So, whether the genes are linked or whether the genes can be recombined, what is deciding this? This is decided by the distance between the genes. Okay, so distance between the genes is the main factor that decides whether the genes are together, that means linked, or genes can have new combinations, whether they can recombine. And when does all these things happen? During meiotic phase, during meiosis. So linkage and recombination. And the basis for this is distance between them. When the distance is less, linkage is more. When the distance is more, recombination is more. Simple thing. When the distance is less, they are linked. When the distance is more, they are more recombined. Recombination frequency will be very high. If they are in two different chromosomes, imagine here I have taken 3 characters, wing size, body color, eye color, all of them are in X chromosome, but their distances are in different areas, isn't it? That is why combination linkage areas are different. But imagine if I am going to take characters which are there in different chromosomes. Some in X chromosome, some in Y chromosome, some in some other autosomes. Then they are independently moving. Isn't it? One is not at all concerned with the other one. They are never linked. They are in different chromosomes. So when they are in different chromosomes, they can go independently or they will follow the law of independent assortment. So why Mendel could get that 9:3:3:1 ratio? Because all the 7 traits that he had taken were in 7 different chromosomes. That was his luck actually. He didn't know anything regarding chromosomes, genes, nothing. But whatever characters he had taken were in different chromosomes. So he could simply come to conclusions very easily. And he could get those data's credibility also he could prove there with mathematical calculations. It was very simple for him to do that. But here Morgan could not get that 9:3:1 ratio because they were not in different chromosomes, same chromosomes. And every time when characters was changing, what was happening? The frequency was keeping on changing because the distance between the genes were different. So, if you have more parental type, means genes are very close or they are linked. If they have more recombinant type, genes are far away. They can have new combinations. So it's all the distance that matters. So, if we can find out the parental combination or recombinant percentages, we can actually work out the distance between the genes. We can find out whether one gene is with another gene or are they far away. We can find it out. So we can actually find out the location of genes on a chromosome. That is, we can map out the genes. We can map out the chromosomes. This was actually done by a student of T.H. Morgan, the student Alfred Sturtevant. He worked on this particular cross, on the results of these crosses. He worked on these percentages and he actually put forth the first genetic map. And based on his ideas only. Even now we have actually done the Human Genome Project, where we could map where in human body each gene is present, where in that chromosome each gene is present, in which chromosome the gene is present, where in that chromosome the gene is present. And children, all these things can actually solve or can pave way for solutions for many genetic disorders. So regarding Mendelian disorders or genetic disorders, gene mapping and all, we shall see in the next video. Thank you for now.